Hi, my name is Irene Roller, and today I'm going to present my work titled Drug Perturbation of Crosstalk between PI3K AKT mTOR and RTK ERK signaling pathways via mathematical modeling. The phosphoinositide 3 kinase protein kinase B mammalian target of rapamycin or PI3K AKT mTOR signaling pathway. It's essential to many physiological and pathological conditions, such as cell proliferation, angiogenesis, metabolism, differentiation, and survival. Stimulation of this pathway enhances growth, survival, and metabolism of cancer cells. This pathway interacts with other signaling pathways such as TGF-beta-SMAD4 signaling pathway, mitogen-activated protein kinase pathway, and WNT beta-catenin pathway. In this schematic diagram, we can see that AKT can directly phosphorylate FOXO and keep it in the cytoplasm to prevent its binding to the promoter of P27 and P21, thus blocks the TGF-beta-SMAD4 mediated cytostatic signals. In this other scheme, we can directly see how the PI3K AKT mTOR signaling pathway interacts with the receptor tyrosine kinase extracellular signal regulated kinase or RTK ERK pathway. PI3K phosphorylates the 3-hydroxyl group of phosphoinositides to produce phosphatidylinositol 3,4,5-triphosphate, which is a second messenger that signals through AKT to activate mammalian target of rapamycin. RTK ERK pathway converges with the PI3K AKT mTOR pathway and is now recognized as an alternative in mTOR activation. In parallel, membrane-associated estrogen receptors binds to PI3K and activates molecules such as AKT and RAS, crosstalking with the growth factor signaling pathways. It's for this reason that we have simulated the effects of mTOR, AKT, and PI3K inhibition on the system's dynamics of phosphoinositide 3 kinase protein kinase B mammalian target of rapamycin signaling pathway coupled with receptor tyrosine kinase extracellular signal regulated kinase pathway. We have used a simplified mathematical model of both signaling pathways crosstalk, introducing in the system terms corresponding to inhibitors of the pathway: everolimus, uprosertib, voxelicib, acetad-8055, GSK 690693, and buparlicib. For the dynamic modeling of the signaling pathway, it was required to obtain the experimental values of the kinetic constant of each pathway reaction, which describe the conversion rate of one component into another in time. The values of the inhibition constants of the drugs were obtained by bibliographic review. With that information, we were able to perform the mathematical models by using ordinary differential equations. They were written using Python programming language syntax. We analyzed the variation of the pathway components using the Ranch-Kara numerical integration algorithms of Tellurium program. Schematicizing the signaling pathway, we could see that the PI3K AKT mTOR signaling pathway is interrelated with RTK ERK pathway through the FOXO component. We can see that PI3K inactivates FOXO indirectly by activating AKT, while AKT does it directly. The obtained simulations show that under increasing mTOR inhibition, RTK ERK pathway is basically not perturbed. This behavior is not replicated in the case of AKT and PI3K inhibition. Steady-state dose-response simulations showed that this effect is mediated by FOXO, because its higher expression caused by AKT inhibition results in an enhancement of RTK activation. In addition, we can deduce that the RTK concentration increases slowly when using the inhibitor buparlisib because it has a low inhibition constant. In the literature, it was found that FOXO both supports and suppresses metastatic breast cancer progression and hence are not classical tumor suppressors. FOXO activity is actually required for proliferation and dissemination of cancer. Moreover, overexpression of FOXO3 is associated with poor prognosis in patients with triple-negative breast cancer, glioblastoma, and gastric cancer. In conclusion, it's possible to hypothesize that cellular context which cause changes of FOX expression may influence greatly the degree of resistance to AKT inhibitors and PI3K inhibitors. We would like to extend our gratitude to Vicerrectorado de Investigación y Posgrado de la Universidad Nacional Mayor de San Marcos for the financial support and to Instituto de Investigaciones de la Amazonía Peruana for the facilities provided. We also want to thank the organization of this event for giving us this opportunity to present our work.